Hi everyone, once again, here is the Cancer Philosopher, and I just want to do a brief video on what exactly is cancer. This was only, there was a couple pages in the book spent on this, defining what exact cancer is. It's part of the last chapter that I'd say has most of the relevant facts to the project that I'm doing, to the course in general. After this, part 6 is more about, it's titled The Fruits of Long Endeavors, and it talks a bit about the nice parts of cancer research that are done today. Some of this will come towards the end of the final presentation, but this part right here, towards the end of part 5 of the book, I felt was really important because it discusses what exactly is cancer, and the defining facts of cancer are sort of what's the focus of the field today. So I thought that was something really important to delve over, and that's why I'm including it here in a separate video. So first, what exactly is cancer? According to the book, The Emperor of All Maladies, there were these two scientists named Weinberg and Hanahan, and they tried to explain the core behavior of more than 100 different types and subtypes of tumors. Now, the question was very audacious in its expansionists, and the answer was even more audacious in terms of its economy. If we look right here, there are 6 different things that cancer was composed of. And to quote them, they say, quote, "We suggest that the vast catalog of cancer cell genotypes is a manifestation of 6 essential alterations in cell physiology that collectively dictate malignant growth." Now, these 6 things were cell sufficiency and growth signals, which means that cancer cells acquire an autonomous drive to proliferate. And they proliferate by themselves, and mitosis just keeps on happening by virtue of activation of oncogenes, like we discussed in the previous video. 2 would be insensitivity to growth inhibitory or anti-growth signals. And that means that cancer cells can inactivate tumor suppressor genes, like we discussed last time. These would normally suppress abnormal growth, but if they don't work as well, then they can't inhibit the growth, and the growth keeps on happening. 3 is evasion of programmed cell death, which is apoptosis, and that means that cancer cells suppress and inactivate genes and pathways that would normally cause the cell to die. If the cell doesn't die, the cell doesn't go through apoptosis, then the cell is going to keep growing. 4, limitless replicative potential. That means that cancer cells activate specific gene pathways that render them immortal even after generations of growth. So they keep growing, and they're limitless, and even though they keep growing, they don't get bad. They can keep growing and growing. They basically don't age. 5 is sustained angiogenesis. Angiogenesis means it talks about refers to the ability of cancer cells to acquire the capacity to get their own blood, to draw out their own blood by taking blood and blood vessels and creating their own blood vessels in order to get blood. And that is what's known as angiogenesis, so sustained angiogenesis. 6, the final one, is tissue invasion and metastasis. And that means that cancer cells must acquire the capacity to migrate to other organs, invade other tissues, and then colonize those organs in order to result in spreading the cancer throughout the body. So those are the 6 things that are involved in cancer. Now, time for the proof. How exactly do we prove that something is a cause of cancer? These 6 things right here are what cancer is. Now, how do we determine what is the source, the root of cancer? So now, in order for something to be the root of cancer, what was agreed upon is that it has to fulfill 3 things. It has to, 1, be present in the diseased organism. That means if the organism has cancer, the organism has to have this mutation, this extra gene, the lack of this gene, etc. So whatever this problem is that we're saying is the cause of cancer has to be present in the diseased organism. Number 2 is that it has to be capable of being isolated from the diseased organism. So a main reason for that is, of course, empiricalism. In order to prove that something like this is the cause, you have to be able to isolate it from the system and then test it separately. And then 3 is you have to be able to recreate the disease in a secondary host when transferred from the diseased organism. And that means that if you take out this problem and you put it into another organism, then it would also get the disease. So now a big problem within research was oncogenes. Oncogenes, of course, fulfill the first 2. Oncogenes are, of course, present in patients who have cancer. And number 2, they can also be isolated. Like we talked about in the last video, Drea tried to clone out this specific oncogene. So they can be cloned out, and oncogenes fulfill the first 2. But the third, no one had shown any sources to this. And that was a big problem because no one could show that you could recreate the disease in a secondary host. That would mean taking out the oncogene and moving it, which you can't really do. But in fact, what happened later in history is that you can in fact induce oncogenes. We talked about X-rays, for instance, and if you recreate the disease, you recreate those oncogenes, then in fact, yes, you can get another diseased organism. And that's why oncogenes are, in fact, one of the causes to cancer.